Okay, aloha. Welcome to lecture 6, which is the second half of chapter 5. Last time we talked about gastrulation and the formation of the notochord and neurulation, and how the presence of the notochord is the primary inducer of neurulation, and then the formation of the neural tube, neural crests, neural folds, and the differentiation of the neural tube, neural crest, and then the surface ectoderm. So today we're going to talk about somites, right? Somites. So while the notochord and the neural plate are busy forming, the mesoderm is still busy proliferating, and it starts to form a really thick region here near the center. This is called the paraxial mesoderm. It also forms an intermediate mesoderm and a lateral mesoderm. Sorry, those are labeled on this side. Near the end of week 3, this paraxial mesoderm starts to segment off into these structures called somites. A somite is roughly the shape of a triangular prism, and it will form a distinct raised elevation on either side of the embryo. In fact, early on, one of the ways that you can tell how old the embryo is by how many somites are present. You can actually count them. The book doesn't talk about it, but each somite actually is subdivided into 3 separate tissue sections. These are called the myotome, the sclerotome, and the dermatome. And if you look at the names, you can kind of guess which ones or which kinds of tissues are derived from each of those tissue sections. The sclerotome being skeletal tissue, and the myotome being muscle tissue, and the dermatome being skin tissue. So collectively, these somites will form most of the axial skeleton and most of the muscles and skin of the back. Okay? And at the same time that the somites and the neural tube are forming, we also start to see these little spaces forming inside the mesoderm. And these little spaces are called intraembryonic coelemic spaces. These coelomic spaces, are going to get larger, and they're going to, guess what, join together, coalesce together, and they're going to form one big space called the intraembryonic coelom. The presence of this intraembryonic coelom is also going to separate the lateral and intermediate mesoderm into a somatic layer and a splanchnic layer. Somatic layer being closest to the ectoderm and the splanchnic layer being closest to the endoderm. Now, this also gives rise to these 2 kind of distinct tissues of layers of tissue. It kind of subdivides the embryo. So when you take the somatic intraembryonic mesoderm and combine it with the ectoderm, those 2 layers of cells together are called the somatopleur. When you take the endoderm and combine it with the splanchnic mesoderm, then it forms the splanchnopleur. It's not going to be super important on the quiz, but it's, just so that you have the information. Now, meanwhile, this embryo here, he's getting bigger and bigger and bigger. And he's reaching a point now where diffusion isn't really effective enough at transferring nutrition. So, in effect, he's getting hungry. He needs to have a more efficient means of transporting nutrients. So the answer to this is called vasculogenesis. Okay, now vasculogenesis, in case it shows up on a quiz, because it might, vasculogenesis is defined as the formation of a rudimentary cardiovascular system. So the formation of a rudimentary cardiovascular system is vasculogenesis, right? Now then, the extraembryonic mesoderm begins to proliferate, and it begins to differentiate, and it forms these little structures, all these little bubbles, are called blood islands, okay? Blood islands are made out of angioblasts. That's the type of cells that they differentiate into is angioblasts, and they form these little clumps of angioblasts called blood islands. What happens is these blood islands begin to hollow out. And as they hollow out, they're going to join together with other hollow spaces from other blood islands, and they're going to start to form this network of tubes. Now, the cells that are near the middle, as this thing begins to hollow out, they're going to do one of two things. They're either going to become flat and form the endothelium of the blood vessels, or they're going to differentiate into hold on, it'll come to me. They're going to differentiate into hemangioblasts. That's what they're called. I had to cheat and look at my notes, actually. I couldn't remember. So they're going to flatten out. They're either going to flatten out and become endothelial cells, or they're going to differentiate into hemangioblasts. And hemangioblasts are going to eventually become blood cells. They're the progenitor cells for blood cells. So hemangiogenesis only happens in the umbilical vesicle, allantois, and the chorion at this stage. It's not happening, and it'll happen in other places during other times. But for now, all the blood is being formed just in those three places: the umbilical vesicle, allantois, and in the chorion. So something you're going to need to know for the quiz, so write it down, is that the inner part of the blood islands, the cells on the inner part of the blood island, will either form endothelial cells or hemangioblasts. They will become one of those two things. Now, once you have these systems, this network of tubes, what's going to happen is these endothelial cells are going to do something called endothelial budding. And endothelial budding basically means that this endothelium is going to proliferate and punch its way through, and it's going to form a new tube, and that new tube is going to be surrounded by more mesoderm. So it's basically forming additional blood vessels. Once you have these vessels formed, endothelial budding occurs, which forms new blood vessels. And this process is called angiogenesis. Okay, so just so we have our terms straight, the formation of a rudimentary blood or cardiovascular system is called vasculogenesis, right? The formation of new blood cells is called hemangiogenesis. And the formation of new blood vessels is called angiogenesis. That's a lot, huh? At any rate, meanwhile, up here in the cardiogenic area, there are two thick muscular tubes that are forming, and these two thick tubes are going to join together. They're going to merge together into one thick tube, which is the rudimentary heart, okay, the rudimentary heart tube. And it's also going to form the great vessels eventually. So this heart tube then connects with all of the blood vessels that are now present in the embryo, and at about day 21, this heart is going to start beating. Once it starts at 21 days, it never stops again for the rest of that person's life until they die, which is a pretty darn big deal if you ask me. This is actually the very first functional primordial system in the embryo, is the cardiovascular system, right? So the angiogenesis and this happens in the embryo itself, also happens in the connecting stalk and in the chorion. You can't see it on this, but the chorion that surrounds this whole embryo, the chorionic sac, all of that is going to have angiogenesis, and all of those blood vessels are going to connect into one single network that connects into the heart. Okay? Now, while all of that was busy happening, these chorionic villi are also busy proliferating and branching off. This is happening during the third week and also at the end of the second week. these are growing, these chorionic villi are growing and budding and branching and filling up with loose mesenchymal tissue. So at this point, they've branched into secondary villi. As they branch out, the same vasculogenic process is going to happen in them. They're going to form blood cells, networks. It's going to connect with the rest of the embryo. And then they're going to continue branching into tertiary villi. So you can see right here, we've got the secondary villi, and then we're starting to have tertiary villi where these secondary villi are beginning to branch. While this is happening, the cytotrophoblast cells gradually, they start to proliferate and surround the chorion, forming a shell around it, right? These guys right here, they're forming a shell around it called the cytotrophoblastic shell, right? And basically this is going to serve to anchor the chorion into the endometrium of the mother. It's going to, and eventually that's going to become the placenta. So basically this cytotrophoblastic shell is going to serve as an anchor for the placenta. And what's going to happen is these tertiary villi are going to branch through the shell, and the branches that go through the shell are called stem or anchoring villi. And then once they break through there, they're going to branch even further out into terminal villi. There's a very small space between the villi and the maternal endometrium, and this is called the intravillous space. And how nutrition gets to the baby or to the embryo at this point is basically mom bleeds into this intravillous space, and then the nutrients and oxygen and other stuff that the embryo needs are going to diffuse through the wall of the villi into the bloodstream, come around, get into the baby's body, and then all the waste is going to be carried back out and diffuse into the intravillous space and then be carried away by endometrial veins, right? So that's vasculogenesis. And that is the end of chapter 5 and the end of lecture 6. So again, if you have any questions or if you need clarification on anything, please make sure that you write down what your questions are or what it is you need clarification on. Bring that with you on Thursday to the Zoom meeting, or if you can't make the Zoom meeting, text me, shoot me an email, set up a personal meeting with me and we'll go over it. So that is going to be it for chapter 5. We'll see you guys hopefully at the Zoom meeting, and then we'll see you at the quiz. Aloha.